And that brings us to the considerations for design. What are the considerations for analysis? What are the advantages of good design? And what is the starting process for developing your trial design? This is a key phrase you'll hear time and again in statistical analysis of any field, but particularly of clinical trials. Analysis follows design. The type of design will lead directly to the type of statistical analysis. For example, you can't analyze a two-way ANOVA unless you have a 2x2 factorial design. So I cannot fit a linear model with A plus B plus A times B that creates 4 groups if I don't have all 4 groups. So if I don't have active A, active B, placebo A, and placebo B, then I can't fit placebo, placebo, active, placebo, active, active, active. That's called a two-way factorial design, and I cannot fit the whole process if I don't have all 4 groups. Similarly, if you have a crossover design, if each participant doesn't complete their crossover, you may not be able to use all arms. So if you have a crossover and you happen to run everyone A to B, and people drop out, and enough people drop out, you won't have enough B information. You cannot estimate change without longitudinal measures. You cannot get an odds ratio from a continuous endpoint unless you dichotomize it or trichotomize it. You can't get a rate of change from a Likert scale. So keep in mind that design will lead to the type of analysis. You cannot fix a poorly designed and poorly run clinical trial with top quality statistical analysis. There's a very common phrase in the statistical world that reads garbage in, garbage out. If you bring us bad data, we can tell you what your experiment died of. So do not expect your statistician to be able to fix mistakes in your design. The design needs to be solid before you move forward. Analysis also will not fix bias or large variability due to a lack of controlled data collection. We cannot wish away large variability. The only thing that will impact large variability is an increase in the sample size, but that's an artificial way to force precision. And your sample size is likely fixed prior to you beginning. So you really need to understand the variability that you have in your endpoint, and you really need to understand what biases can come from measuring it. Statistically, we cannot fix biased data. So what about good design overall? Again, from Piantadosi, good design allows investigators to satisfy ethical constraints. It permits efficient use of scarce resources. Good design will isolate the treatment effect of interest from confounders. So if you've paid close attention to the homogeneity and the inclusion and exclusion criteria, the treatment effect that you see should be due to the treatment. Good design will control your precision. It will reduce your selection bias and your observer bias, 2 of the most common types of biases that we see. Good design will minimize and quantify random error or uncertainty. So random error is just that. It's random. It's not controllable. But with good design, you can minimize the impact and assess that impact of random error or uncertainty. Again, you have to do that with good design. You cannot, on the back end of a study after the data has been collected, statistically address your biases or your random error. Good design will simplify and validate the analysis and will increase the external validity of the trial. So if we run this study and we have these results and no one ever is able to reproduce these in any other manner, then the trial is not externally valid. So besides analysis following design and keeping an eye on the statistical considerations for bias and error, what else do we need to think about? Well, you need to think about the number and ratio of treatments. So how many treatment arms will I have? What is the ratio of those treatment arms in terms of participants? So if I already know a lot about the existing treatment and I have a fairly good idea what that variability and outcome will be, do I really need to collect as many people in that treatment arm? If the new treatment arm, you're a little bit more uncertain about it, if you have more people in that treatment arm, you'll reduce the standard error, which helps with your estimation. So perhaps I want 2 times the people in the new intervention to every 1 person in the standard intervention. So think about the number and ratio of treatments. What are the dosing types and administration issues? Every decision that you make will affect your data. Dosing types are really important. If you're asking somebody to take 17 pills in a week, you may see more dropout in that arm from pill fatigue versus, say, 4 or 5 pills a week. If you're asking somebody to take a daily injection versus a weekly injection, you may see more dropout in the daily injection. So you need to be aware of the dosing types. If it is infusions that people have to come in for, if it is surgical, where are the points along the way that you could lose participants prior to introducing the intervention? And where are the points after the intervention is introduced that there would be administration issues? So how frequently do participants need to come back in for their intervention if it's medication? If it is a behavioral intervention, how is it going to be administered? And what are the concerns around that? Equipoise issues. Equipoise is a term that was first introduced in 1987 by Benjamin Friedman. And what it really means is that there is genuine uncertainty in the medical community over whether a treatment will be beneficial. So going into this trial, you must really be unsure, for sure, that your new intervention or treatment will succeed. If there is mounting evidence that the treatment or new intervention in one arm of the trial is significantly outperforming other treatment arms, then you don't have equipoise. You are subjecting people to an inferior treatment when it was not effective. Or the flip side of that is if it becomes very clear that the new treatment is not effective, again, you're subjecting people to certain risks. So you want to make sure that you have equipoise, that you really truly have no idea how this trial will end up. As the investigator, you hope that the new intervention will be more effective, but you really don't know. And this is a really key point to when it comes to finding a statistical group to work with you. You want to work with independent statisticians that have no game in mind. They do not have any concern about what the outcome will be other than to provide you with accurate estimates and determination. So this is why often in large-scale clinical trials that are run by pharmaceutical companies, they seek out independent statisticians to run, manage, and execute those trials. In addition, if a clinical trial is wholly funded by a pharmaceutical company, any major publication of those results will require independent verification of that data being sent to another statistician or statistical group for verification. So in addition to the number and ratio of treatments, the dosing and types of administration, the equipoise issues, you want to have an understanding of the timing and administration of randomization. So when will it occur and how will it occur? Are there any immediate side effects that could pose a risk to a participant? Even if rare, are there allergic reaction problems? Are there cardiac issues? Do you need to watch the participant? Are there endpoints that need to be collected right at the timing of administration? The relationship between the length of study, the endpoints, and the population under study is a very key relationship. Are you hoping to affect something short-term? Are you hoping to affect something long-term? Is this a long-term chronic illness? Is it a short-term acute problem? Your length of study endpoints and the population under study should reflect all of those considerations. So what phase of study should you undertake? These are very clearly defined in the literature, keeping in mind that early phase studies are phase 1 and phase 2. Phase 1 is for safety and tolerability. Is this intervention safe, and can people tolerate it? Phase 2 will look at safety and some efficacy. Confirmatory phases will look at efficacy and safety. Notice the switch in the efficacy coming first and safety coming second. So the confirmatory phase is really to confirm the efficacy and gather additional information on the safety profile. And then the post-marketing phases, phase 4 studies, are long-term safety and efficacy. All of these considerations, the trial goals, the good design, the considerations, and the phases, aim towards simplicity. We are striving for parsimony. There are several definitions for parsimony. Economically, it's extreme unwillingness to spend money or use resources. And that's actually the case for clinical trials as well. Clinical trials also operate in the definition of scientific parsimony, where the scientific principle that things are usually connected or behave in the simplest or most economical way is what we're going for. In statistical terms, parsimony is trying to get the best prediction with the least number of predictors. In all of those instances, and in clinical trials, we do not want to overcomplicate matters. Don't pick a neat design for the sake of completing the trial. Just because a 4-arm crossover adaptive design, placebo-controlled, with an active comparator arm seems really neat, you may not need that to estimate the effects and the magnitude of those effects that you're looking for. So simply picking a neat design complicates everything thereafter. Remember, choosing the design is one of the first things you do, and it will set up everything else that you do, from operations to analysis to your results to patients in the clinic. So pick the simplest design to answer your question at hand. That doesn't mean that the simplest design for you isn't a complex design, but it needs to be justified, and it shouldn't be done just because it's new and exciting. Don't forget about blinding or masking. So how are you going to blind or mask participants and or their observers, or both? And the design of your study should fit your objectives. So think about simplicity, think about meeting your goals in the simplest fashion. There is always the temptation when you have a participant sitting in front of you in the clinic to measure as much data as you can. The problem with that is it leads to trial fatigue. You get dropouts, you get people who will stop coming back, and in some cases, you have data that you will never look at. If the primary trial results are equivocal, that is, if you find for the null hypothesis, oftentimes you'll see interest in the study data wane. And then you've collected all of this information that may never be used. Or funding runs out and there's no one to finish the analyses. So keep in mind your design about only collecting the information that is critical to address the question or questions at hand, and then keep your design as simple as possible.